Hello students, welcome to Molecular Biology Part 11. The concept is protein synthesis. Introduction. Proteins are giant molecules. Proteins are biological importance because they are not only forming the building material of the cell but acts as enzymes controlling all the biochemical reactions going on in the living system and also control the expression of phenotypic traits. Chemically, proteins are polymers of amino acids. The amino acid molecules are linked together in a linear fashion, forming a polypeptide chain. The adjacent amino acids are joined together by covalent peptide bond between the carboxyl group of one amino acid and the amine group of the other amino acid. All the proteins which are required by the cell are synthesized by the cell itself. Protein synthesis includes arrangement of a definite number of amino acids in a definite sequential order. The sequence of amino acids in the polypeptide chain is determined by the sequence of nucleotides in the polynucleotide chain of DNA. The next topic is transcription. The process of copying genetic information from one strand of DNA into RNA is termed as transcription. In the process of replication, the total DNA of an organism gets duplicated. In transcription, only one segment or only one part, one segment of DNA and only one of the strand is copied into RNA. So just now we have discussed the ultimate expression of gene is protein synthesis. This already we discussed. So protein synthesis is ultimate action of a gene, and we are all very well known that amino acids are made up of polypeptide chains. Polypeptide chain made up of, and this polypeptide chain is formed by amino acids. Amino acids. That means the peptide bond is formed between the amino acids. So in the polypeptide chain, the amino acids are arranged in a sequence. First amino acid bond with second amino acid. It is bond with third amino acid and it is bound with fourth amino acid, like that. The n number of amino acids present in a polypeptide chain. This sequence of amino acid, the sequence of amino acid in a polypeptide chain is determined the sequence of nucleotides present on DNA. So the sequence of amino acid is decided by, it is decided by or determined by sequence of nucleotide or nucleotides present on DNA. So where this DNA is present? So this DNA present in nucleus. It is present in nucleus. And we are all very well known that where the protein synthesis takes place. So the synthesis, the protein synthesis takes place in ribosomes. Takes place in ribosomes. And where these ribosomes are located, the ribosomes are present in the cytoplasm. Okay, so protein synthesis required amino acid sequence. That sequence information present in DNA in nucleus. Then how that information will comes to this ribosomes? That is the topic now. So how that sequence of amino acids present on DNA come to the ribosomes. That means enters into the cytoplasm. So now for that, the first step, the sequence present on the amino acid sequence present on DNA is copied onto the RNA that we will call as the transcription. That RNA brought that amino acid sequence to the ribosomes to synthesize a polypeptide chain in a protein. So that is the information given here. So the process of copying genetic information, nothing but that sequence of amino acid information from one strand of DNA into RNA is termed as transcription. Okay. So now in this transcription copying, DNA is double helix. DNA is a double helical in structure. In this double helical structure, only one strand, in these two strands, only one strand is involved in the copying of that information onto the DNA. Now, we previously we studied about replication. So in this replication, the entire DNA molecule duplicates into two new DNA molecules. Whereas in transcription, only one segment of DNA is copied onto the RNA. Why are both the strands of DNA not copied during transcription? So just now we said that only one strand of DNA is involved in the transcription, and in that only a segment of DNA is copied or transcribed into a RNA. So why both strands are not copied during transcription? So let's see. If both the DNA strands act as template, that means involved in the transcription, if both the strands are involved in transcription, they would code they would code for RNA molecule with different sequences, and in turn they code for the different proteins. That is the first problem. So if both the strands acts as a template, that means acts as a parent strand to copy down to the RNA, they code for RNA molecule with the different sequences. and in turn they code for different proteins. Next, the two RNA molecules, if produced simultaneously, would be complementary to each other. The two RNA molecules become complementary to each other. So we are all very well known that one strand, so DNA, it is a double helical in structure. So now in this, if both these strands are involved, this strand as well as this strand, both these strands are involved in the transcription. What happens? It is produced one RNA and it is produced another RNA, two RNA. Now see, these two strands are complementary to each other. The two strands, this DNA and this DNA, both are complementary to each other. Then if the both these strands are produced RNA, that two RNA molecules, new RNA molecules, also complementary to each other. Hence, they would form a double stranded RNA. So due to the complementary to each other, then the two RNA molecules are coiled and form as a double stranded RNA. This will prevent RNA from being translated into protein, and the transcription will not occur. When the RNA become double stranded, the transcription will not occur and that RNA is not translated as a protein. The transcription unit. So what is meant by a transcription unit? The segment of DNA on which one primary transcript is transcribed is known as transcription. The primary transcript in prokaryotes is generally equivalent to the mRNA molecule. In eukaryotes, the primary transcript must be processed to form mRNA. I think you can understand these 2 points. I am repeating again. Whatever the primary transcript formed in prokaryotes that directly acts as a mRNA. But in eukaryotes, the primary transcript which is produced from the segment of DNA must be processed. It need to undergo process to form as a mRNA. It is not directly form as mRNA. It form as a heterogeneous nuclear mRNA. Heterogeneous, that is before mRNA. In eukaryotes, the RNA form as hnRNA, heterogeneous nuclear RNA. This hrNA is processed and form as mRNA. We will discuss later how it is, which one is called as hnRNA. HN, heterogeneous nuclear RNA. It is a first primary transcript formed in eukaryotes and it undergo processed and it undergo further changes to form as a mRNA. That changes and all we will discuss during transcription in eukaryotes. Now, in eukaryotes, the transcription unit mainly consists of three regions in the DNA. The transcription unit consists three regions. They are promoter, structural gene, terminator. The structural gene is present in the center, while the promoter and terminator regions are present at the two ends of structural gene. Suppose if it is a structural gene, the promoter gene as well as terminator gene present at the two ends of that structural genes. Let's see that a promoter. Promoter is present at the beginning towards the fifth end of the structural gene. So first point, it represents its location where it is present. So promoter is present at the beginning towards the 5 end of the structural gene upstream. I will show in the diagram. Promoter is the site on the DNA to which an RNA polymerase enzyme molecule binds prior to initiating transcription. So, what is this promoter? Promoter is a site on DNA. What happens? At this site, promoter site, RNA polymerase enzyme binds to initiate the transcription. So, the promoter is a site where the transcription initiation will start. Now, promoter also contains the information that determines which of the two DNA strands will be transcribed, not only initiating the transcription, the promoter also play a key role. So DNA having two strands. In the two strands, the two strands, which DNA strand will be transcribed, will be copied. That information is also present in the promoter. Based on the position of promoter, template and coding strands are defined based on the position. If the position of promoter and terminator are interchanged, then the definition of template and coding strands will be reversed. In this case, the original coding strand will now become template strand. So, the template and coding strands are defined based on the promoter position. If that promoter and terminator positions are interchanged. So, for example, so this is the structural gene. Okay. So now this is the a promoter here. It is a promoter. Clear. And this is the terminator one end. This point already structural gene one end is terminator, another end it is a promoter is present. So now what we are saying, the position of promoter defines the template as well as coding strands. So here structural gene, it is nothing but the structural, that is a DNA strand only. It is a promoter, terminator and structural gene, they are present on DNA. It has two strands. For example, it is a template T and this strand is a coding strand. Now, when these two positions are interchanged, that means if it is present here and if it is moved to this region, that means how we can represent so now this is a structural gene structural gene, and now we are changing that, interchanging. That means this is a promoter, and then it is a terminator. Clear? When these positions are interchanged, now in this case, the green color strand, it is a template. Now it is defined, it become reversed. That means this one become template and this one become coding strand. So in this case, the original coding strand will now become template. Original coding strand is this one. When it is interchanged, that will become the template. I think it is clear. Let's see. This is the representation of this transcription unit. So what we said, promoter is present on the upstream region. This is the upstream, this region, and this is the downstream region. So this promoter is present on the fifth end of that coding strand. Okay. Now what we said, the promoter is the initiates the transcription, and at this site RNA polymerase enzyme initiates that transcription. Clear? And this promoter and terminator present on the two ends of that structural gene. Okay, this point is also, and not only initiating that, this promoter position decides the template and coding strands, and also the promoter having the information about which strand of these two need to undergo transcription. That is also, and finally we are given when the positions are changed, the promoter and the terminator positions are changed, then coding and template strands become reversed. Okay, so now see here the upstream region. Here in this is the upstream of a gene, and on fifth end, so see here 5 dash. Don't compare here. Here it is first strand, it is 3 dash, but it is reversed. Okay, so now this 5 to 3, that means it is considered as non-template. That means here in the green, this one coding strand does not involve, coding strand not involve, not involved in transcription. Remember it, transcription does not involve in the transcription. Coding strand, generally coding strand we will imagine that it is involving the transcription. No, template strand is always involved in the transcription, so it involve in transcription. It involved in the transcription, remember it. So non-template strand or maybe coding strand, both are same. Non-template, that means the strand which is not acts as a template. Template is a strand which is involved in the copying or transcription. Okay, so now template strand, that means it is involved in the transcription. Non-template strand, it is 5 to 3. So it is a coding strand what is labeled. This one is the non-template. It is a non-template and it is a template. Okay, so we are saying that the upstream based on this non-template strand. So promoter having 5 end. This region, that means sorry, coding strand having 5 end. At this region we will call it as the upstream gene. So at this upstream, this promoter is initiates. So it is the site of promotion, that means promoter, and it is also a site of initiation of transcription. And transcription initiates from the promoter and it is moved towards the terminator region downstream. Okay, the next part of transcription unit is structural gene. Although only one strand of DNA is used in transcription by convention, the structural gene is defined by both the strands of DNA. As the two strands of DNA have opposite polarity, that means one strand is 5 to 3 direction and other strand is 3 to 5 direction. 3 to 5 direction. Now, the DNA dependent RNA polymerase, this is essential amino, that means enzyme involved in the transcription. DNA dependent RNA polymerase enzyme catalyze the polymerization only in 5 to 3 direction. Only in 5 to 3 direction. So the strand that has polarity 3 to 5 acts as the template. It is called as the template strand. Remember that. DNA dependent RNA polymerase catalyze the polymerization only in 3 to 5 to 3 direction. 5 to 3 direction. Why? Because third carbon having hydroxyl group, that is easy to join the nucleotides. Okay, so now here 3 to 5, that means this strand is acts as a template because it is opposite polarity, no, newly formed RNA having 5 to 3 direction. Okay, so that's why this acts as a template strand. So this coding strand, it does not act as a template strand. So during mRNA formation, that means transcription, from this template strand, coding is copied onto the RNA in 5 to 3 direction. The other strand DNA that actually does not participate and has polarity 5 to 3 is referred as a coding strand. It does not participate in transcription. That's why it is also called as non-template strand. The coding strand is displaced during transcription. So we are all very well known that again to formation of a new strand to parallel to this template strand, here the hydrogen bonds between the base pair should be broken. That means unwinding is essential. So that's why it is displaced with the RNA strand. Coding strand Coding strand does not code for anything in transcription process. However, all reference points while defining a transcription unit is made with the coding strand. The RNA transcript would have the same sequence of nitrogen bases as on the coding strand. You may understand this one. So clear, see, two strands we are giving the two different colors. So this is a template strand. So it is 3 to 5 direction. 3 to 5. Now another DNA strand. So another DNA strand, it is a separated one. Suppose during transcription, it is 5 to 3 direction. It is another DNA. Now it is template now. it is a template DNA. That means the sequence. These are two are separated. Actually, both the DNAs are complementary to each other. That means here A, G, C, T, A, G. These are the sequence present on this strand. Now opposite strand having complementary. That means A bonds with T. It is DNA, no? T, and C. It is G. It is A, and it is T, and it is C. This is the sequence. Remember the sequence on coding DNA. Coding DNA strand. Okay. What is that sequence? T, C, G, A, T, C. This is the sequence. Remember it. So now what we are saying that these are separated and new strand, that means RNA, is formed. This is displaced. They are unwinded and a new strand is formed parallel to the template strand. New RNA strand. Now A is bound with an RNA. So A is bound with, we know RNA instead of T what is present? U is present. So here U is present. G, C, C, G, T, A it is bound with U and G is C. That means the newly synthesized RNA strand, if you are observe the sequence on RNA, first one is U and second one is C, third one is G, fourth one is A, and fifth one is U, and last sixth one is C. Once compare the sequence present on RNA and sequence present on coding DNA. So second code is C. Here also C. Here it is G, G, A, and the last C and C. But here it is T because it is DNA instead of uracil, thymine is present. It is RNA instead of thymine, uracil is present. That means the coding present on the coding strand DNA as well as the sequence present in RNA is similar. So that is mentioned. We'll have the same sequence of nitrogen bases on the strand, that is a coding strand. The whatever the newly synthesized RNA having same sequence which is present on the sequence of this coding strand DNA. Suppose a hypothetical sequence from a transcription unit on a structural gene is given below. See here, a sequence is given on this 3 to 5 direction. That means it is a template strand. This is T template strand. So this is the sequence given on that. And now this 5 to 3 direction, it is a coding strand. It is C coding strand. So template strand, coding strand, two strands is given here. The base sequence of RNA transcribed would be complementary to the template strand. Being RNA, it will have uracil instead of thymine. So now when this template strand is copied, when the template strand is copied onto the RNA, so during transcription, this is displaced with RNA. New RNA strand is formed. Let's see RNA, which sequence we can see on the RNA. RNA sequence. RNA sequence. See, first one is A. A is bound with T, but thymine is absent. Then uracil U. Next T is bound with A and G is bound with C. Next C is bound with G, A is bound with uracil, T is bound with A and C, G, U, A, C, G. So I wrote up to here. So this template strand, when it is copied onto the this part, when it is copied onto the, that means copied as a RNA, this is the sequence. Now observe that sequence up to here now in the template strand. So now instead of what we are mentioned being RNA, it will have uracil instead of T. So first one is T. Here it is RNA, no? That's why it is uracil. Next A. So here it is A and A. Next C, G and thymine, uracil is present. Next adenine, cytosine, guanine, uracil and again adenine. So CG, that means C and G. So next we are not written, that means we are taken up to here only, G only, not T. So if you are take the remaining 5 letters also, then it will come like that. So whatever the RNA it is transcribed from the template strand, the sequence of nucleotides present on the transcribed RNA is similar to the DNA, coding DNA strand. The third part of transcription unit is terminator. Terminator is present at the end of the structural gene towards the third end with respect to the coding strand. So remember that the terminator is present towards the third end with respect to the coding strand. So we are mentioning the sites of promoter as well as terminator based on the coding strand. So what is the specialty of this coding strand? Coding strand cannot involve or does not involved in transcription. Does not involved in transcription. So terminator is at the opposite end of the promoter. It normally defines the end process of transcription. So terminator indicates the end process of transcription. The gene on the transcription unit. Gene. It is defined as basic unit of heredity or functional unit of inheritance. So gene is defined as basic unit of heredity or functional unit of inheritance. The gene is DNA sequence that controls the synthesis of tRNA or RNA molecules. These two tRNA and rRNA involves in the formation of protein synthesis. Cistron. A cistron refers to a short segment of DNA that has complete information for the formation of polypeptide chain is called as cistron. I am repeating definition again. The short segment of DNA that has complete information for the formation of a polypeptide chain is called cistron. In other words, a cistron codes for a polypeptide molecule or RNA molecule is called a cistron. Polycistronic. Several genes are transcribed on a single mRNA. It occurs in bacteria or maybe prokaryotes. Several genes transcribed on a single mRNA is called polycistronic, and these polycistronic genes are occurred in bacteria or maybe prokaryotes. So all these structural genes transcribed as a single mRNA, and this single mRNA translate from the different types of proteins. So all the information of these several genes, they are present on single mRNA. Based on that, several proteins are formed in prokaryotes. Monocistronic. Each gene is transcribed on a single mRNA called monocistronic. Each gene. Polycistronic means several genes are transcribed on a single mRNA, and monocistronic, a single gene transcribes on a single mRNA. So these monocistronic genes are occurs in eukaryotes. The mRNA transcribed from monocistronic gene is called as the monocistronic mRNA. So a monocistronic gene is not a continuous straight sequence of DNA, but it has a stretches of non-coding DNA as well. So a monocistronic gene, not a continuous, it consists both coding regions as well as non-coding regions. So let's see. So from this structural gene, every gene form as one mRNA, and that one mRNA form as a single protein. It is different from the polycistronic. All genes are formed as a form transcribed on a single mRNA and form several proteins, that is polycistronic. Whereas in monocistronic, a single gene transcribed on a single mRNA, it forms as a single protein. Exons. So just now we said that in eukaryotes, in monocistronic gene, whatever the mRNA is transcribed, it is consists of both the coding as well as the non-coding regions. Coding and non-coding regions. Now, exons. Exons means the coding sequences on monocistronic gene is called as the exons. So see here the red colored one. These regions, the colored regions are considered as exons. They are coding sequences. They code a amino acid or maybe a nucleotides. They code for a particular amino acid. Clear. So coding sequences on monocistronic gene is called exons. These coding and non-coding sequences are absent in prokaryotes, and in prokaryotes only exons are present. That means coding sequences are present continuously, but the coding sequences are not present continuously in monocistronic gene. Now introns. So the non-coding sequences on monocistronic gene is called as introns. So these regions are called as the introns. So introns are not code for any amino acid. That non-coding sequences are called introns. So in polycistronic gene, polycistronic gene, polycistronic gene. Only exons are present, means all are continuous coding sequences. There is no introns. Introns are absent in polycistronic gene. So what are introns? Introns are the non-coding sequences. Non-coding sequences are called as the introns, and these are the coding sequences. Exons are coding sequences. Both the introns as well as axons are present in monocistronic gene. So in starting we said no, in eukaryotes the mRNA need to undergo process. The transcript unit, primary transcript, need to undergo process to form mRNA. So this is the process. What is that? First it form the heterogeneous mRNA and it undergo the splicing. Splicing means these introns are cut and rejoins the continuous. That means these introns are cut and joins these axons form as a continuous strand. Then it forms as a mRNA. So that process occurs before forming mRNA is that removing that introns and joining the axons. That process takes place to form mRNA in eukaryotes. This is not required in prokaryotes because all the strand consists continuous coding exons, but in monocistronic gene that present in eukaryotes, it does not consists a continuous coding sequences. It is consists non-coding sequences also. The non-coding sequences are called introns and coding sequences are called as the exons. So during transcription, the primary transcript in eukaryotes undergoes a process to form mRNA. That process is to remove these introns to form a continuous strand.